Welcome to this explainer. Have you ever looked at your own hands and thought of yourself not as flesh and bone, but as billions of lines of code? Well, today we're diving into something absolutely mind-bending. We're stepping into an era where biology is literally becoming a programmable language. You know, exactly like the software running your laptop or your smartphone right now.
Seriously, think about this for a second. What if curing a disease was as easy as updating your phone? Imagine a world where, instead of going through months of really harsh, exhausting chemical treatments, fixing a faulty gene was just like downloading a software patch while you sleep. I know, it sounds like pure science fiction, but thanks to some wild new breakthroughs combining artificial intelligence and biology, this is becoming a very real possibility.
Which kind of makes you wonder, what if we've been looking for medicine the wrong way this whole time? Because for decades, our whole approach to discovering drugs has basically been scavenging through nature. We find random molecules that plants or bacteria happen to make, and we cross our fingers trying to turn them into human medicines. But researchers are realizing this is pretty flawed. Why? Well, because we've been relying on nature's accidental designs, and those designs were never actually meant to cure human diseases in the first place.
So, let's dive into Section 1, Nature's Fragile Molecular Machines.
To really understand why this old way of finding drugs is hitting a massive wall, we need to look at the tiny workers inside our cells, proteins. Think of nature's proteins like a cheap, highly specialized used car. They are fantastic at doing the one exact job evolution gave them, but they are incredibly fragile. If a scientist tries to tweak that natural protein, say upgrade the engine so it can fight a human disease, it just immediately breaks down. Every little change destroys its stability. But AI-designed proteins? They're like a heavy-duty, custom-built truck chassis. They're robust, they're super resilient, and they are built from the ground up to handle massive, heavy-duty upgrades without falling apart.
Which brings us to Section 2, upgrading the starting line. So instead of starting with those fragile natural proteins, scientists are now using AI to build way stronger starting points. In the lab, they call this having biological runway. Basically, biological runway is just the structural strength a protein needs to survive a bunch of artificial changes without completely collapsing. Researchers are using an AI called Protein MPN to redesign these proteins. Now, the AI doesn't necessarily invent the perfect finished drug on day one, but what it does is give scientists a stabilized, heavy-duty foundation. With that strong base, scientists can make hundreds of tweaks to perfectly target a disease, and the whole thing won't just fall to pieces.
There's a quote from the source material that absolutely hits the nail on the head for me. It says, AI may never become the machine that hands us a finished drug. It may become the machine that gives drug discovery better ancestors. The real magic of AI in the lab right now isn't that it just magically spits out a finished pill. It's that it gives researchers a super robust, flexible platform that they can then evolve into actual life-saving cures.
Okay, let's look at Section 3, from Botox to ALS. Let's look at exactly how powerful this new approach is in the real world, using some groundbreaking work from David Liu's lab. Step 1, the researchers started with the botulinum neurotoxin enzyme. Yeah, literally the active ingredient in Botox. In nature, it's a deadly toxin that acts like molecular scissors to cut proteins. Step 2, they used that AI we mentioned, Protein MPN, to upgrade its structure. They made it way more stable without changing its core cutting ability. Step 3, because the AI gave them all that biological runway, they ran it through rapid tests in the lab to basically reprogram it. Their goal? To turn this Botox enzyme into precision molecular scissors designed to hunt down and cut a very specific toxic protein that causes ALS or Lou Gehrig's disease.
And the result? 79. The AI redesigned enzyme was 79 times more specific at targeting that ALS-causing protein compared to just using the natural enzyme as a starting point. When researchers tried to upgrade the natural version, it just failed. It fell apart. But the AI-stabilized version, it absolutely thrived, hitting a level of precision that could genuinely lead to a groundbreaking therapy for a truly devastating disease.
Let's zoom out a bit for Section 4, the biology operating system. We've been looking at a single protein, but now we're looking at the entire genetic code, all thanks to a monumental new AI model called EVO2. I just want you to take a second and absorb this number. 9 trillion. EVO2 was trained on 9 trillion DNA letters. That is the exact, mind-boggling amount of biological code this massive AI has ingested. It's basically learning the fundamental language of life on a scale we have literally never seen before.
See, older AI models were mostly just trained on simple bacteria. But EVO2, its training data covers practically all domains of life. It read the DNA of over 100,000 species. We're talking animals and plants, bacteria, and even viruses.
It's acting like a universal Google Translate for biology. It understands the deep genetic grammar that connects a tiny single-celled organism all the way up to a complex human being like you and me.
Because of this massive training, Evo 2 has unbelievable processing power. It can read 1 million DNA letters all at once. That huge perspective lets the AI see the whole big picture. It's like reading an entire chapter of a book to understand the plot, rather than just staring at a single sentence. Evo 2 can see how completely distant, seemingly unrelated parts of our genetic code actually interact with each other to control how our bodies work.
Which leads us straight into Section 5, a programmable medical future. So what does this actually mean for you and your healthcare? Well, we are already seeing real-world impacts. Right now, Evo 2 can analyze mutations in the BRCA1 gene, that's a gene heavily linked to breast cancer, and it can predict with over 90% accuracy whether a specific mutation is totally harmless or if it's dangerous. It's literally turning clinical genetics from a highly stressful guessing game into programmable, predictable certainty. This is going to massively speed up how quickly patients get life-saving diagnosis. When you compare how we used to do things with where we're going, the shift is just unbelievable. Past medicine was all about discovery, a whole lot of trial and error, and trying to force fragile, natural proteins to do things they didn't want to do. It was basically a lot of guessing and just hoping for the best. But future medicine? It's defined by active design, AI prediction, and upgradable genomes. We are officially moving away from accidentally stumbling upon medicines in the wild to actively engineering precise, targeted therapies on a computer screen.
We are heading straight toward a reality of precision gene therapies that have zero side effects. Why? Because they're programmed to only turn on inside 6 cells, leaving all your healthy cells completely untouched. We are talking about upgradable biological machines living inside our bodies. Ultimately, we're gaining the ability to read and write entirely new DNA from scratch. Imagine downloading a custom-designed biological patch that is tailored just for your body. It sounds like magic, but honestly, it's just the new reality of biological engineering.
So it leaves us with one massive question. If biology is just code, what will we program next? As artificial intelligence learns to fluently speak the language of our DNA, the limits of medicine aren't bound by what nature accidentally provided anymore. They're only bound by what we can imagine and design. Thank you so much for joining me on this explainer, and I really hope this leaves you just as fascinated and curious about this whole generative biology revolution as I am.